Welcome back, dear students. Let's continue with biofertilizers. And now we're going to talk about the symbiotic nitrogen fixation, especially with respect to rhizobium. As we all know, rhizobium occurs as a symbiotic organism associated with the root nodules of leguminous plants. Now, this is a picture of the root nodule of leguminous plant, and you can see the nodules. And associated with these nodules are being found to be certain bacteria, and they are referred to as a rhizobia. This is the scanning electron microscopic image of the bacteria, which has been shown here. And you can see that when you consider rhizobia, they mainly include different types of bacteria. They could include members of the alpha proteobacterial genera, including members like Allorhizobium, Azorhizobium, Bradyrhizobium, Mesorhizobium, Caenorhizobium, and simply Rhizobium also, which we together call them as rhizobia. Now, they all do the process of nitrogen fixation, and that's how they add fertility towards the soil. As I told you, biofertilizers can act as two ways, either by nitrogen fixation or by phosphate solubilizing. So in the case of rhizobium, they add fertility to the soil with the help of nitrogen fixation process. Now, if you go to look into a normal environment, not always rhizobium is invading the leguminous plant. In a nitrogen starving environment, of course, the plant will need the rhizobium's association. But when nitrogen is sufficient enough, the plant need not associate with the rhizobium. So, rhizobium, it normally lives freely in the soil, and what happens when the soil is being found to be nitrogen sufficient, the plant will produce certain ethylene compounds which will inhibit the nodulation. Nodulation is a process where nodules are being formed. Okay, and when the nitrogen starved soil is being formed, what happens? The rhizobia will have what? The rhizobia will try to invade the particular plants. Now, even when the process of invasion has to be carried out, there do exist some interaction that has to be happened between the what? The the leguminous plant and the rhizobia. Now, in the movie Rhizobia Avatar, you can see that the hero is trying to tame his particular flying bird. Okay, there is an interaction, and you can see that in order to tame it, he has to fight along with it, and the final interaction or the connection between the hero as well as his bird which he uses happens only when an interaction happens between both of them. The same way here in the case of rhizobium, what happens as well and the plant, there exists a something which is being found to be inhibiting the invasion of rhizobium. The plant will produce certain various compounds such as superoxide radicals, hydrogen peroxides, as well as N2O and all that, as well as homoglutathione and glutathione, which will normally inhibit the growth of rhizobium or will prevent the infection process. And rhizobium in turn will have to produce some antioxidant defenses. And when this antioxidant defenses becomes sufficient enough to overcome the oxidative stress which is being given by the plant, only then an infection process can be initiated. So there is a fight between the plant and the rhizobium also here. In the case of here, the compounds that we use, of course, is the oxidative stress. So the rhizobium, if it has to invade a leguminous plant, it has to overcome this what? Oxidative stress. Now, once the oxidative stress is okay, fine. If it has to enter, it has to overcome the oxidative stress. But at the same time, if a plant is living in a what? Nutrient starving environment, okay, it will or a nitrogen starving environment, specifically speaking, it will produce certain compounds called the flavonoids. Okay, these are the flavonoids. These flavonoids normally they are in fact they get induced and they go and attached onto the rhizobium. And this is a type of signaling what is happening. And once this flavonoids go and attach to the rhizobium, in the rhizobium also certain what certain genes get activated. And what happens, an interaction happens, or the attachment of this bacteria onto the what onto the root hair cells of the plant do happen. And the interaction is normally happening with the help of lectin and ricardescence. These are some proteins, okay. On the plant surface there are certain lectins, and on the surface of the rhizobia there are present certain compounds such as the ricardescence. So that interaction happens by certain proteins over here. Okay. Now, once such interactions happen, the nod genes, okay, like this rhizobium also will start getting activated. So, in the case of, as I mentioned here, for the initiation of this particular association, certain signaling molecules have to be transferred between the plant and the rhizobium. And the plant produces these flavonoid inducer molecules, which will go and attached onto a NOD gene. Okay, it's been found in the bacteria, and what happens, the bacteria will get activated and it produces a, in turn it will produce a nodulation factor signaling compound. Okay, that is, okay, there's a gene in the bacteria called the NOD gene that gets activated, and the bacteria in turn will produce another compound called the NOD factor signaling compound. Okay, and what happens is, now once this NOD factor signal has been produced by rhizobium, the plant cell will get it. And in the plant cell, what happens, the intracellular calcium will be found to be increased. And what happens, when the NOD factor as well as this calcium is being increased, when the calcium spiking happens, the root hair cells of the plant will start to curl. Okay, and this curling will in turn help the what the bacterial cells to get entrapped also. And the NOD factors thus will trigger the root cortex cells to reinitiate the cell division, and the cells will eventually form a nodule primordium that will affect the invading rhizobium. You can have a look at this in this particular picture. So the flavonoids are produced by the particular plant when the nitrogen starvation happens, and it goes and binds to the rhizobium, and in the rhizobium it activates the NOD genes, and at the same time the bacteria gets attached onto the rhizobium gets attached onto the what? The plant cell with the help of ricardescence, which are these are proteins found on the plant, not on the plant, on the bacteria, and on the plant there are lectins. So that happens, and once that is being activated, the NOD genes is being found, found in the rhizobium gets activated to produce, and they produces a NOD factor. And the NOD factor comes here, and also the increase of calcium happens in the particular plant, and the root hair deformation and the bacterial attachment happens. This is the first process, like what the series of events which would initiate the attachment of rhizobium onto the plant cell. Thereafter, again we have certain processes which are being needed. You can see that then again the bacteria has to penetrate into the root hair cell, and infection thread growth should happen. And this, so the curling of the root hair has been happening, and the bacteria is coming here. So the bacteria comes in, and an infection thread has been formed here because the bacteria gets entrapped. Then after that, the growth of this infection thread is mainly coordinated by the nucleus. Okay, and what happens? The infection thread is growing inside towards each other as a nucleus has been passing, and what happens, the release of these bacteria inside into the host cell normally happens. And once it is inside, Okay, you can see that the formation of bacterioids happen. That is, the bacteria is being surrounded by a membrane, and a symbiosome is also being formed over here. So this is a process by which the rhizobium extends from one cell to the other, and as a result it's a cell to cell transfer, and first then finally what happens, a root nodule is being formed over here. So what we have to say is, once the nod factor happens, the bacteria produces a symbiotically active exopolysaccharide, and this will change the plant cell wall, and it will invaginate. The plasma membrane will start invaginating, and it could lead to the development of a bacteria-filled tube-like structure called the infection thread. That's called the infection thread. And the plant cell division derives the growth of the infection thread, and upon penetrating the base of the root hair cell, the bacteria will stimulate the growth of the infection thread through the additional plant's cell walls as well as the membrane. And at the same time, you can see that plant hormone cytokinin will also act along with the nod factor during the plant cell mitosis. And finally, the infection thread filled with the bacteria reaches the inner plant cortex, where the bacterial cell is endocytosed, as we saw in the previous picture, by a membrane, and it will form a symbiosome. And if it is here that each bacterial cell differentiate into the nitrogen fixation form called the bacterioid. And see, it is in this, each cell is referred to as a bacterioid. So here what we have to say is, once the curling happens, the infection thread has been formed, and this infection thread goes again further instead, infecting all the cells, and we find that I told you the plant hormones are also being formed, and so more growth happens over here, and then nodules are also, that is associated with the root hairs, and in the regions where the are being infected by the rhizobium, what happens, root nodules are being formed. And this is how the process of rhizobia based establishment happens in a leguminous plant. Hope so we were, you are clear with it. So I guess it's clear. So mainly what we have to speak is, an interaction or a signaling happens between the plant as well as the bacteria, and it is that which initiates the rhizobium to attach onto the leguminous plant. And always this is being found to be in fact specific. A rhizobium can attach onto not all plants. It is specific, like only in the leguminous plants it can affect. So this is possible only if the interactions are being found to be correct. So with this we come to an end of rhizobium based nitrogen fixation. Of course enzymes and all that I will be explaining it in next session. Thank you for now.